Hi, my name is Emily Pinlac, and I'm a sophomore in the School of Engineering majoring in chemical and biochemical engineering. This summer, I've had the privilege of being able to conduct research in Dr. Benjamin Schuster's lab in the Chemical and Biochemical Engineering Department at Rutgers, studying biopolymers and their applications. The work that I'm presenting today is a culmination of these efforts. My project focuses on developing a protocol for conducting a technique known as passive microrheology on biomolecular condensates. Passive microrheology relies on the latent thermal energy of a substance to measure its viscosity through the tracking of microscopic probes diffusing through a small volume of sample. Our lab's interest in this technique is its ability to be applied to biomolecular condensates, specifically in measuring the microrheology of intrinsically disordered proteins, which phase separate due to weak multivalent interactions. But why even measure the microrheology of these condensates to begin with? We needed a streamlined method of this technique for an upcoming project that aims to characterize the sequence determinants of microrheology for intrinsically disordered proteins. It's also worth noting that understanding the rheology of biomolecular condensates would aid in elucidating the impact of viscosity changes to various important biological functions, such as cellular processes and even the development of some neurodegenerative diseases. There are two components to the protocol we developed: data collection and data analysis. Data collection involves wet lab work in the form of proper sample preparation for microscopy. The ability for our protocol to successfully collect reliable data relied on two main factors: ensuring that the beads were in a location amenable to tracking, that is, not at the edge of the condensate, and ensuring that the condensate was in a form that was amenable to gathering reliable data. These two challenges were intertwined, as you're about to see. Tracking the particles in the condensate while it was in droplet form was problematic for several reasons. Droplets are more prone to extraneous translational motion that would impact the reliability of resulting data, and often contained less particles to track per video. Particles would also sometimes gather at the border between the condensate phase and the dilute phase, otherwise known as the interface. To address this, we decided to instead allow the condensate to gather at the bottom of the well as one continuous layer. This gathering process eliminated much of the unneeded bulk motion that we were observing, and also allowed more of the particles to exist within the condensed phase by decreasing surface area. We also accelerated this process by centrifuging the samples post-plating, allowing more of the condensate to pellet in the bottom of the well through centrifugal force as opposed to waiting for gravity to pull the condensate downwards. This proved to be especially effective in increasing the amount of data that we could gather per experiment, as it resulted in a thicker film of condensate, and thus more particles eligible for tracking. It also helps to ensure that more particles would be inside the condensate instead of at the interface. The results of our experiments were a collection of videos per sample. For data analysis, we used Trackpy, a particle tracking package for Python that allowed us to determine the ensemble mean square displacement of each sample through our data. From there, the viscosity can be calculated using the Stokes-Einstein equation. We found that centrifugation has no significant effect on condensate viscosity. It is also worth noting that centrifugation of samples allows for the maximization of the amount of particles that are able to be tracked, as it increases the thickness of the condensate layer. Unfortunately, there are limitations to the reliability of our experiments due to our sample concentration. Our anticipated concentration of each polyion for our model system was 60 micromolar polyK and 1.5 millimolar UTP, but due to improper storage of our aliquots, the effective concentration of both components are different than anticipated for most of our experiments. Therefore, our viscosity measurements were off from the literature by a factor of around 1.8. Our next steps involve troubleshooting a surface chemistry issue that arose with another sample, the RGG domain found in the protein LAF1 that we used in the early exploratory stages of this project. We noticed that there was very little partitioning of the nanoparticles into the condensed phase due to the unfavorable reactions between the carboxyl groups on the nanoparticles and RGG. We hypothesized that covalently attaching polyarginine to these carboxyl groups, thus covering up the RGG-phobic regions, if you will, of the nanoparticles, would make partitioning more favorable. I would like to thank the Douglas Residential College for providing the support and resources necessary to participate in this project. I would also like to especially thank Dr. Schuster for his help and guidance, as well as for his continued support of my research interests.